Okay, so the experimental plan which we are going to discuss. Yesterday we came to a point that we should collect the blood sample in a population. Then we should isolate DNA, and then we should compare the gene sequences, because in the databases which are available, biological databases, we know what is the normal gene sequence. I was citing the example of beta thalassemia. Beta thalassemia, basically, there is a blood disorder which generally occurs due to a mutation in the gene which is coding for hemoglobin protein. I can cite another example of sickle cell anemia. Let me admit some of the people who are there. Okay, so sickle cell anemia, I have already explained this picture. You people are familiar with this. Here, let me have a pointer. This is the like part of the uh, sequence for hemoglobin gene. So at this position, normal hemoglobin gene consists of CTC, and this particular genetic code is responsible for glutamic acid amino acid in the protein. When a mutation is there is only one mutation for the sickle cell anemia. Okay, only one mutation. So when this T is changed to A, so now it becomes CAC. This genetic code is different now from CTC, and it is going to code for a different amino acid, valine. So these two amino acids are totally different from each other. Glutamic acid, it is a polar amino acid. Valine, it is a hydrophobic amino acid. So in that way, the protein structure is going to change, and ultimately hemoglobin is abnormal. It cannot transport oxygen now. So here the plan is like what we are going to discuss: how to uh, how to find out the known mutations. The technology which we are going to discuss today, three four parts are there in that particular experimental strategy, and we will be able to tell whether somebody is having this particular mutation or not. And if mutation is there, what is the situation, whether it is going to be, let me explain. I should have a pen. So normal case is at the both the chromosome the person is going to have this. So we can say TT, that is going to be the normal, right? Normal individual here at this particular position, here also T, and in the second chromosome also we will be having C. So we can say that TT is the genotype of a normal individual. On the other hand, when the disease is going to be there, it is going to be AA, and in the case of heterozygous, it is going to be AT. Okay, so the technology which we are going to discuss, it is going to tell us all these three things. First of all, mutation is there or not, right? It is, and the individual is going to be homozygous or heterozygous. All these questions uh, will be answered. Only thing is, only the restriction to the technology which we are going to discuss is mutation should be known, similar to like sickle cell anemia. We want to work on sickle cell anemia, and this is a very well-known mutation. So we can do the screening by using the technology which we are going to discuss today. So that's why I have given another title, that detection of known mutations for a particular genetic disorder. Okay, so let's start. So first step is the collection of blood sample. Second step is genomic DNA isolation from those blood samples. Then what should be our third step? Can you think?
Sir, comparing the gene sequence.
No, it should be the last one. Like before, first we have to get that particular gene.
Sir, cell lysis we have to do.
Yeah, those things are over now because uh, the second step, which I just told, that is the genomic DNA isolation.
Genome.
Right? For genomic DNA isolation, definitely you have to do the cell lysis and removal of the protein, removal of RNA, all those things, and ultimately you have the DNA. DNA is too big. Like I told, 20. We have 46 chromosomes in total.
3 into 10 to the power 9 base pairs.
Right, 3 into 10 to the power 9 base pair. We have to now only out of this genomic DNA, which is too big. Our interest is on a single gene. So what should we do? This is the genomic DNA. And here our interest is, let's say here. So what we have to do?
Sir, I don't think there is a way to directly go at that particular point. So we have to read. So we have to first create a mechanism to actually know which base pair it is. Then we
Okay.
have to read the entire length. Actually, we have to do because again, pinpointing is not possible. But when we are close to that part, we can do like a search, and then we can come to that point.
Okay. So I will just modify a little bit what you are saying. Once we are done with the genomic DNA isolation, this is the entire genomic DNA, and our interest is on this region. So we have to get only this region now. We are not interested in rest of the part here. Let's say I'm working on sickle cell anemia. I will be interested only in this hemoglobin gene. I'm not interested in rest of the genomic DNA. So the idea is to get that particular gene, only that particular part of the chromosome. Let's say I'm interested here. So I have to get this. And the technology which we are going to discuss, those techniques also require this particular part in good amount. Okay. So we have to basically isolate this particular gene in good amount, in good concentration or good amount. It is the idea. Then we can work on that particular gene. We can we can work out. So these are the three techniques. One is called gel electrophoresis. Second is polymerase chain reaction (PCR), and last one is RFLP. Once we are done with the genomic DNA, we have to make sure that there are two things. Once, let's say, we have isolated the genomic DNA, there are two things which we have to do. We have to make sure that really we have isolated the DNA. It's yellow color, it's too faint. We are done with the genomic DNA isolation. We have to make sure that yes, we have isolated DNA. Make sure that we have, yes, DNA is there in the tube. That is the first thing. And second thing here, we have to quantify this DNA. Okay, there are two important points. Whenever we do the genomic DNA isolation, it is a must to see that DNA. Yes, we have seen the DNA, that means it is there. And now quantify how much DNA do you have? Because reason behind the quantification is like these two particular techniques, they require a particular amount. Let's say 10 nanogram of DNA. I'm just giving an hypothetical example. Let's say PCR, it requires 10 nanogram of the DNA. So we can add 10 nanogram of the DNA in the tube if we know what is the concentration of our DNA sample. Okay. So today, these are the two things which we have to do in parallel. Gel electrophoresis, it is the technique to visualize DNA on the gel. Okay, gel electrophoresis, it is the technique to visualize DNA on the gel. So it will tell us, yes, DNA is there in the tube. Okay. Second thing, quantification. So first we will go to, I will skip these slides. I will go to the quantification part. Then we will come to the agrogel electrophoresis. Why to quantify nucleic acid? Reason is very clear that the PCR or RFLP techniques, they are going to use a particular amount, which I cited, like 10 nanogram or 100 nanogram. So it is a must to know the concentration of your nucleic acid, which is DNA here. How we are going to quantify DNA? We are going to use one of the physiochemical property of DNA or nucleic acid is that sugar phosphate backbone does not contribute to absorption. Absorption is there. DNA actually can absorb the UV light at 260 nanometer. Not only DNA, RNA also. DNA and RNA both can absorb the UV light at 260 nanometer. What are the different molecules which are making DNA or RNA? Can you name? We have seen the structure of DNA, right?
Sir, as in phosphates and that,
Yes.
phos
Okay.
adenine,
Okay.
guanine, cytosine, thymine.
Yes. So we have three things there. One is sugar, second is phosphate, and third one is the base, AGTC, adenine, guanine, cytosine, or thymine. So this absorption of the UV light is mediated by the bases, AGTC. Sugar and phosphate, they are not going to contribute anything with respect to the absorption. So this is a very basic property that these are the adenine, guanine, cytosine, and thymine. These are the bases, purine and pyrimidine bases, which absorb the light at 260 nanometer very precisely. So we are going to use an instrument which is called spectroscopy. Have you seen this instrument? Like if you have taken the chemistry course, maybe Dr. Ghosh, he has shown the spectroscopy or spectrophotometer.
No, sir.
Okay.
Computer science students.
Okay. So Dr. Shirododi Ghosh, he has not taken any course till now for you people.
No, sir.
Okay. Okay. No worry. So let's go through the spectrophotometer instrument. So this instrument can tell you how much of the light is absorbed. Basically, this instrument is going to tell you absorbance, AB, absorbance of light by a particular sample, whatever is. It is not necessary to be DNA or RNA. If you have any solution and you want to see how much light uh, can be absorbed by this, you can use the spectrophotometer. Or like you have different samples where different colors are there, even the intensity of that color can be quantified by the spectrophotometer. So there are two things, transmittance and absorbance, and based on that we will be able to tell the concentration. This is the simple schematic of a spectrophotometer. Light source is there which is going to produce polychromatic light. Then a wavelength selector is there because we have to set the wavelength here. We want the absorbance for DNA or RNA at 260 nanometer very precisely. So wavelength selector is there, and once we fix that we want light at 260 nanometer, it is going to produce the monochromatic light. Then a sample holder is there where we can fit our cuvette, which is having the DNA or RNA sample, and the DNA or RNA sample is going to absorb the light, and ultimately we are going to have the transmitted light. And here detector is there with a readout, and we are going to get the absorbance value, that this much light is absorbed. This is the idea. Okay, so now because here using this spectrophotometer, what we are going to get, we are going to get absorbance, and what we need a concentration. So there should be some relationship between these two things, absorbance and concentration of nucleic acid. Okay, so let's go to that particular point, and it is called Beer-Lambert's law. Beer-Lambert's law, these are the name of the scientist. They concluded that there is a linear relationship between the absorbance and the concentration of the absorbing species. Or simply it can be written like this: A is equal to ABC, or in some books it is written like this: A is equal to epsilon, that is the extinction coefficient for a particular molecule. L is the path length, like here you are putting your DNA sample in a sample holder or cuvette. So what is the path length here? That is L. And in most of these spectrophotometer, this L is equal to 1 centimeter. Okay, and C, that is the concentration. So if we know the absorbance, we know the extinction coefficient, we know the path length, we can calculate the concentration of any sample, not only DNA or RNA, any biological sample. So this is one thing. One formula is A is equal to epsilon LC. Okay. Second formula is there is a relationship between the transmittance and absorbance. Transmittance is given by the equation I by I naught. Maybe you can note down these formulas. We are going to solve some of the numericals. And the relationship between the absorbance and transmittance is A is equal to minus log T, where T is I by I naught. So ultimately you can remember this formula, that A is equal to minus log T, or log 10 I naught by I is equal to epsilon LC. You can note down the extinction coefficient for double stranded DNA, it is 0.020, and for single stranded RNA, it is 0.025. Let's see this particular question. Here, this question is with respect to RNA. Similar concept is same for DNA also. Let's consider that volume of RNA sample is 50 microliter. That means finally you have your final volume in which your RNA sample is there, that is 50 microliter. And for the quantification purpose, what you have done, you have taken 10 microliter from here, 10 microliter of the original sample, you mixed with 490 microliter of distilled water. So now this volume becomes 500 microliter, right? And absorbance at 260, where path length is given, it is 0.75. So what should be the RNA concentration? Can you do this calculation?
Sir, concentration. Sir, can you do this once?
Yes, why not? So we know that I will derive the like the formula which directly we can use. Okay, so Beer-Lambert's law is A is equal to epsilon LC, right? So if you want to know the concentration, the formula for the concentration will become A is equal to, sorry, C is equal to A upon epsilon into L. So concentration should become absorbance. Here, what is the extinction coefficient for RNA? Did you note down?
0.025.
Okay, so 0.025, path length is 1 centimeter. So the formula which we can remember for the concentration of RNA, it will be absorbance at 260 into, what should I write here? 0.025. So 40, right? 40. And because here we are diluting our
But
sample.
over here, by A of 260, we are saying at 260 nanometers, the
Wave,
wavelength of light.
yes,
Okay, sir.
yes,
Okay.
right.
Got it.
So, and here we have to consider our dilution factor because we are diluting our sample, right? So this is going to be the final formula you can, you should remember, which is derived from the Beer-Lambert's law. The concentration of RNA will be A260 into 40 into dilution factor. Dilution factor means how much fold, sorry, how many fold you have diluted your sample. How to calculate the dilution factor? Very simple. Like here, you are taking 10 microliter of the RNA sample, mixing with 490 microliter of water. So what is the volume now? 500 microliter. So 500 divided by the sample which you have taken, it is 10. So it will be 50 here. That means you have diluted your sample 50 times and then you are measuring the absorbance. So the concentration of the actual sample, original sample, will be absorbance at 260 into 40 into your dilution factor. And that makes sense. So now can you calculate what should be the concentration?
Sir, a small question I had. Sir, we
Yes,
already got the value of C below. How did we get into dilution factor?
dilution factor means like here, what you are doing, whenever we do this DNA or RNA concentration, original sample is 50 microliter and that is having very, very high concentration. So what we do,
Yes.
we take a small amount from there. Let's say here I have taken 10 microliter, and according to the, like, different types of spectrophotometers are there. Some spectrophotometer, they need minimum 500 microliter sample. Okay.
Yes, sir.
Some spectrophotometer, like they require at least 100 microliter sample there. That means we cannot use our entire 50 microliter there, because here, let's say, this in this particular question, that spectrophotometer requires 500 microliter of the sample. So we cannot dilute our sample entirely. So what we are doing, we are just taking little bit, 10 microliter, mixing with 490 and making it 500. So whatever absorbance you are getting, that is of the diluted sample. So the concentration, whatever you are getting, that again is going to be for the diluted sample. And what we
Okay.
actually want to know, concentration actually here of the original RNA sample.
Yes, sir.
So
Got it.
that's why we have to
Yes.
multiply by dilution factor.
Sir, 1,500.
Yes, what should be the unit there? So unit will be according to this extinction coefficient. We should remember the unit here. So if you consider this, it is going to be, your concentration is going to be microgram per ml, right?
Yes, sir. Microgram per ml.
Microgram per ml. So now can you tell me what is going to be the general formula? Like this is the formula, C is equal to A260 into 40 into dilution factor. It is the formula for RNA. So similarly, can you derive what should be the formula for DNA?
Yes, sir. Sir, A into 50 into dilution factor.
Yes, you are right. So these are the formulas which we have to remember, and we should know the reason behind these formulas, from where we are getting. We are getting these formulas from the Beer-Lambert's law, considering the extinction coefficient, and especially the units, microgram per ml. So when suppose we are getting OD of 1, so simply we can use the formula and we will know if OD is 1, that means, and dilution factor is also 1. So in that case, the DNA concentration is going to be 50 microgram per ml, which is written here. For RNA, it is going to be 40 microgram, or if you have single stranded very small oligonucleotide, it is going to be 33 microgram per ml. Okay, so now let's discuss some more problems based on the concentration and Beer-Lambert's law. Everything is clear here. Okay. So apart from the concentration, there was second part of the question. What is the total amount of RNA? So total amount of RNA will be like we have 15 microgram per ml. That is the concentration. And what is the volume? It is 50 microliter. That means 0.050 ml. So that means we have 75 microgram of RNA in our sample. Okay, here we have 1, 2, 3, 4. These are 4 good questions based on the Beer-Lambert's law. Anybody's done with the first question?
Sir, just checking the units actually.
Okay. For the first one, you can express your answer in micromolar.
Sir, 80.3 I'm getting for first one.
Yes, 80.3 micromolar. Yes, that is the right answer.
Sir, we will get a 0.19.
Yes, 0.19.
No units. No units.
Yes, with no unit.
Sir, 90%. Sir, I'm getting basically 10 I naught is equal to I for fourth. So that I don't know how to convert. Basically 90%, but I don't know absorbed or transferred.
Okay, so let's see this particular question. So basically, the relationship was A is equal to minus
Minus 10
log A.
I by I naught. Yes, sir.
So here, uh, optical density is 1. That means absorbance is 1. Okay, so minus log T is equal to 1. That means log t is equal to -1. So t will be 10 to the power -1. That means 1 by 10. If we convert this into the percentage, so t will be 10%. Transmittance is 10%. That means 90% of the light is absorbed.
90% of the light is okay, sir. Got it.
Right? Yes. C is the answer.
Sir, 60%.
No, recheck your calculation. Any change in the answer?
Sir, I'm trying once.
Okay.
Okay, sir, not getting.
Okay. So let's try this. Okay. So keep calculation. So what I'm going to do? I'm going to have a white screen and we can have this question there. Okay, so now let's try. Beam of light passes through 1 centimeter of a colored solution. So here L is 1 centimeter. 80% of the incident light is transmitted. 80%. T is 0.8. Okay, so if the incident light passes through 2 centimeter of the same solution. Okay, same solution means concentration is going to be the same. Here, this is L1, here L2, it becomes 2 centimeter. The percentage of the transmitted light. Okay, so we know that A is equal to basically,
64.
sorry,
64.
64, yes.
Oh, okay,
64.
sir. I did calculation mistake. Sir, actually,
Yes.
right now when you wrote 0.8, then I got that answer. I was actually taking
Okay,
0.2. That is why I was getting wrong.
okay, okay, okay. Yeah. Okay, so now we are done with the first thing which we should do after the genomic DNA isolation, the quantification. We have gone through some of the problems. So now we have this expertise. Okay. Now the second thing is we have to visualize DNA also. And this technique is called agarose gel electrophoresis. So agarose gel electrophoresis, it is a technique which can separate the molecules according to these properties: size, charge, and shape. If the structure is very compact, very highly folded, it will run through the gel very fast. If the structure is not very compact, that means it is going to move slowly when we are applying an electrical field. What is the charge on the DNA? Positive or negative?
Sir, I think no charge is there.
No, charge is there because at the 5, like you have the sugar phosphate backbone. Phosphate group is there. So,
Yes, sir.
so negative charge is there. At the 3' end, you have OH minus. 5' and you have again phosphate group. And entire backbone is also of the phosphate, sugar phosphate backbone is there. So overall, DNA is negatively charged molecule. Same thing for RNA. RNA is also negatively charged molecule. Okay, so DNA, when we are going to apply the electrical field, imagine that you have a gel. Let's say here this is the gel, and here some well is there. In that well, you have loaded your DNA sample, and here let's say it is the positive charge. Sorry, it should not be here positive. DNA should be loaded towards the negative side, and here it should be the positive one. So when you apply the electrical field, DNA is going to move in this direction. Okay, so when this DNA actually has run approximately 3/4 of this gel, let's say when your DNA actually reaches here, you should visualize. Otherwise, what will happen if you have just loaded the DNA sample, you apply the electrical field, and if so, if you have applied this electrical field for a long time, what will happen? This DNA will go out of the gel also. So we have to be very careful in this technique that DNA should be within the gel. Then only you will be able to visualize. Otherwise, if you have you have exceeded the time, the DNA will be gone out of the gel because electrical field is there, and here buffer is there in which this gel is sitting. This gel is sitting basically in a buffer, because we need a buffer here so that your electrical current can pass, right? So if the time is more, then DNA will be there in the buffer, then you will not be able to visualize. And it's very important that always load the DNA sample at the minus end so that DNA can move towards the positive charge. Okay, so now second proper, like size. Let's say I have a mixture of DNA. I have a mixture of DNA. Some of the DNA fragments, the size of them is 200 base pair. Some are 500 base pair. Some are or 1 kb, 1 kilo base pair. So what is going to be the order of these? If I run, if I do this agrogel electrophoresis, let's say I have loaded here, there was a well, and in this well I have loaded this mixture. So, and how many bands should I get here in agrogel electrophoresis?
So 3.
So 3 bands will be there. Okay. So what should be the size of this band?
Sir, where is the negative again?
So here it should be the negative pole, and
Okay,
here
sir.
it should be the positive one.
Sir, so I am assuming smallest will move fastest. So this should be 200, then
Yes,
500.
yes, and it should be 1 kb. Okay, so this is the idea. This is very, like, this is one of the most famous technique in biotechnology. Now also, if you go to biotech department and you visit the genetics lab, somebody definitely will be doing the agrogel electrophoresis at present also. This is very routine thing in the biotech lab. So agrogel electrophoresis, basically it is a technique which can separate the molecules according to its size, charge, and shape. These are the three properties which we are going to consider. We have to, so there are some steps. We have to prepare a special buffer. We have to prepare the gel, and then one thing is that how will you visualize DNA? DNA actually is having no color. Once we are done with this agro gel, we have to visualize the DNA. I'm just giving an overview. In the next lecture, we can discuss in detail. There are several things which are required. One thing is gel, buffer, and some sort of system by which we can visualize DNA. So it is going to be a dye, and the dye name is ETBR, ethidium bromide. This is carcinogenic, and it should be handled very, very carefully. What do you mean by carcinogenic? Or it can cause mutation. It should not come into contact of your skin by any mistake. You have to be extremely careful. If a little bit, a very small amount of this carcinogen came into contact of your skin, that means it has caused some mutation in your DNA. So generally, we do not allow the BTEX to touch this chemical. Always, like, only faculty is going to add this ETBR into the agro gel. It is very, very, very harmful chemical, because maybe you will not know the consequence. The next generation, your progeny, they will they will have the consequence. This is too dangerous. But here the property of this ETBR is that it can bind to DNA. It can bind to DNA, and if you put under UV light, upon binding to DNA, it will show the fluorescence. So in that way, you are going to visualize the DNA wherever in this gel. Let's say here, this is the gel, and here DNA is there. So it is going to bind. ETBR is going to bind to the DNA, and if you put now this gel under UV light, it is going to show fluorescence. And in that way, you are going to detect the, yes, I have the DNA. You will be able to visualize DNA here as a band. If you have mixture, several DNA fragments are there. Accordingly, you are going to see those bands, and those bands are getting separated in the agar gel according to their size. the low molecular weight fragment is going to run very fast. Like here, which we have seen, the 200 base pair, it has gone to the bottom. It is going to run fast. It is going to be the fastest here among these three. Okay, so I will stop here. Then we are going to discuss this technique. What I'm going to do, I'm going to upload 2 small videos of agarose gel electrophoresis on LMS. Okay, if there is no size limit, so that you can go through those videos, then we can discuss. Then it will be easy to visualize. Okay. Any
Sir,
doubt?
one thing I
Yes.
wanted to, yes, sir. Sir,
Yes. Yes.
in
Yeah.
this charge, I was asking, sir, you said that phosphate are there, but I was thinking that for charge, there should be, means if there is phosphate, then there is some other also compound which is having positive charge. So why we are giving negative charge to it?
No, I didn't understand your question.
Sir, in like if we have, let's say, salt NaCl and chlorine has negative charge, then sodium has positive charge, then only they are
Mhm.
forming compound, right? So in this
Okay.
also, compound will have positive charge if phosphate is having negative charge. So I was
No.
thinking
Okay. Okay. No. Here in the DNA, when we are doing the DNA isolation, we have to remove the like definitely like salts will be there in the your cell also inside the cell. But here during the DNA, when we are purifying DNA, we are going to remove the salt impurities. So ultimately, when the all this, like let's say Mg plus 2, yes, Mg plus 2 is there in the cell, and maybe it is bound to DNA. You can say that. So in that way, negative charge is shielded. But the DNA isolation protocols, they will remove the salt impurities. There will not be any salt. Finally, when you have the DNA in your tube, in the last step, you will have only DNA in that, no salt. So in that way, you are going to have the negative charge.
oh, okay, sir, got it.
Right? Yeah. But yes, if salt is there, it can shield the negative charge. Like yes, like NaCl or Mg plus 2, they can bind to that phosphate group and they can shield. Yes, that is possible. But here, all in all these protocols we have done, we are taking care of that. Okay, so if there is no other question, let's end the session here, and we will plan our quiz next week on 24th. I will email you the syllabus also, and I will upload the answers like you people have requested last week also, but that was too busy for me. I could not do. But definitely, maybe today or tomorrow, you can find the answers on LMS for the practice questions as well as the assignment. Most probably by the weekend, I will email you people. You people will be able to find the assignment. Only one assignment is going to be there in our course. Okay, so some good questions will be there. Theoretical and numerical, both kind of mixtures will be there, and assignment is going to be there for 50 marks. Okay, and maybe 2 or 3 weeks will be there to solve that, and that time is enough for those questions. Those are not going to be too difficult, but good questions, theory as well as numerical, both will be there. Any doubt with respect to the assignment or quiz?
No, sir.
Okay. Okay, so let's end the session. See you people next week.
Yes, sir. Thank you, sir.
Okay, welcome.